Thank you, Jennifer and Lauren, for this interesting talk. And I would thank you, the International Sunflower Association, for this meeting and to give me the opportunity to present my current work. So I will present you the preliminary results we obtained in the lab on the project Heliopollen, which aims to study the impact of drought stress on sunflowers' attractivity to pollinators. I want to stress out that this project is ongoing and new results are being added daily. Next slide. As a quick introduction, I remind you the consequences of global warming. Southern Europe, which corresponds to the main area for sunflower culture, experience longer and harsher drought periods. In summer, during the sunflower bloom and grain filling, resulting in lower yields. Concomitantly, there is a 25 to 75% deficit in pollination service in many European countries, especially sunflower producing ones. On the other part, the European Environment Agency projects a reduction of water availability of 10 to 40% in Southern Europe by 2100. This results in a projected decrease of crop yields by 15% in Southwest France, Serbia, Italy, and by 55% in Spain, Greece, and Portugal by 2050. The effect of the interaction between pollinators and water availability on these yields remain unknown. During these past years, the Sunrise project, coordinated by Nicolas Langlade, studied the effect of drought stress applied to grain filling in yields. In this present study, we address the question of drought stress applied at bloom stage on attractivity to pollinators to evaluate the yield loss due to pollination decrease. We studied the effect of drought on floral morphology, especially corolla depth, which is known to have a major effect on attractivity, as Gerard showed yesterday. We also studied its effect on pollen production and composition, and finally on nectar volume and composition. We ran the experiments on 3 usual suspects lines identified in a previous project, oleosol, for their differential behavior on yield stability in response to drought stress, which corresponds to the ability for the plant to maintain its reproductive fitness. IR is the most sensitive line, while CI is more tolerant and XRQ is intermediate. XRQ has its genome entirely sequenced and annotated and was published in 2017. IR is also sequenced and is available on INRAE bioinfo facility, but CI isn't yet sequenced. Plants were grown on Heliopollen in Toulouse. Heliopollen is an automated outdoor platform where plants are grown in pots. They have almost field conditions except for water supply, which is monitored by a robot. It weighs the pots, calculates the water availability, and adjusts water to reach a preset value. In our case, 100% of evaporated needs as control, and 20% as the assay. So we evaluated the effect of water deprivation of floret morphology. First, a quick reminder of the anatomy of the sunflower. The head is composed of 50 to 1,000 florets of 2 types, the ray florets on the outside, which are sterile and hold a bright yellow ligule, and the disc floret, which are fertile, sit on top of a single ovary, and the florets have a centripede development. When We then have a single head. We then have on a single head different stages of development corresponding to a time vector, as shown on the right. The younger being closer to the center, and the older ones are closer to the exterior. Just after anthesis, the florets have 1 day of male or staminate stage with antheraise by filament elongation, followed by pollen presentation triggered by style elongation. Then on the second day, the style is fully elongated. The stamens retract and the florets enter female stage for 2 days. When looking closer, we see that the petals are fused in a tube-shaped corolla, and unlike this picture shows, the nectary is at the very bottom of the floret here. It is a polygonal or round structure, round ring, 200 to 350 micrometers high and 450 to 800 micrometers wide. The nectary sits on the top of the ovary. It surrounds the base of the style and is close to sieve elements, as shown by the yellow arrowheads. When the florets wither and fall, the dry nectary remains on the seed hull, which is obvious on this photo of white-seeded variety. The nectary is part of the female apparatus. Since the corolla length is a major trait involved in sunflower's attractivity to pollinators, the effect of drought stress on this trait wasn't explored on sunflower until now, except by Jennifer and Lorraine. Florets were scanned with a regular scanner in 60% glycerol and measured with ImageJ software. When exposed to drought stress, all 3 genotypes experience a slight but insignificant shortening, while genotypic effect is highly significant. So the effect of drought stress on corolla length is unlikely to have an effect on attractivity, but if it had one, it would be a positive one. So let's have a look at pollen, which is the main, if not only, source of lipid, protein, and nucleic acids necessary for brood rearing. Prior to pollen collection, heads are freely foraged by bees and other pollinators, then covered with nets for 24 hours at the 4th day after the F1 stage. Pollen is first weighed and then freeze-fractured. Then lipids are extracted with acetone followed by methanol chloroform. And then protein content is assayed by the standard Barford method according to the protocol published by Evans in 1991. So the pollen quantities or 3 lines have very different pollen release. CI produces as few pollen as 1 milligram per head and per day, while IR produces 40-fold more. And when exposed to drought stress, CI and XRQ have a strong 25% decrease in pollen production, while IR has only a milder 16% decrease. On the other hand, the lipid and protein content do not significantly vary in response to drought stress and is not significantly different between our 3 lines. So the strong drought effect on pollen production is likely to have an effect on attractivity, but not the lipid and protein composition variations. The other floral product being foraged is, of course, nectar. It contains sugar, mainly glucose, fructose, and sucrose, that are the only source of energy for adult bees. We estimated attractivity on a 17 hybrids trial by the average of 9 instant counts between 12 and 8 AM and this was repeated on 3 days. The predominant species is honeybee, followed by bumblebees and mega achilles. We also had diptera, but a very low frequency. On this panel, attractivity is significantly correlated with nectar volume, which is not a surprise and has already been shown on other species. But this also confirms the genetic variability on nectar production and attractivity index. On our lines, while CI had a very low pollen production, it has the largest nectar production of all 3 lines. And IR, which has the greatest pollen load, has the lower nectar production, while XRQ is still intermediate. They also have a different behavior in response to drought stress, where IR and XRQ have a strong nectar flow decrease of around 45%, while CI's decrease is half. This nectar loss dynamics goes in the same direction than grain yield loss, which suggests a common female function stability between nectar production and seed set. When looking to the solute concentrations, it appears that IR doesn't only has the lower nectar volume, but also solute concentrations, while XRQ and CI have much higher concentration and even higher than mean concentration already published. Concentration decrease in response to drought stress is also very different between our 3 genotypes, with a 30% concentration decrease for IR, a milder 20% decrease for XRQ, and a stability for CI. When sticking these results together, CI produces 200 micrograms sugar per floret, while irrigated, with a 30% decrease with drought stress. IR produces only 20 micrograms sugar per floret and has a big 60% decrease. And XRQ produces 160 micrograms sugar per floret with also a big 60% decrease. So we saw that nectar volume and concentration vary according to genotype, but also sugar composition. While IR and XRQ have glucose and fructose in equivalent amounts with only traces of sucrose, which is in accordance with previously published results, CI has 50% sucrose. Waller in 1972 showed that bees prefer sucrose over fructose and glucose, and Femme de Leg in 1985 showed that in sunflower, attractivity is correlated with the presence of sucrose in nectar. The sugar composition does not vary with drought stress. The relative sugar proportion in IR and XRQ strongly suggests that a cell wall invertase is involved in sucrose inversion, but the glucose-fructose sucrose ratio in CI rather suggests a spontaneous sucrose inversion. So we raised the question of the involvement of cell wall invertase and if CI lacks this cell wall invertase. In a recent paper, Minami et al. Showed that in Chinese cabbage, inversion is mediated by C-WinV4, cell wall invertase 4, and leads to an increased osmotic pressure, driving nectar increased volume by passive osmotic diffusion of water. In sunflower, we have 7 copies of C-WinV4. 6 copies are disseminated throughout the genome, corresponding to the exemplification showed in Baudouin et al. In 2017, plus local duplication on chromosome 1. do CI really lack all these 7 copies? It's much, it appears unlikely. The other question are all 7 copies expressed in XRQ, IR, and the gang? It seems more probable that only few copies, maybe only one, is active in XRQ, and CI only lacks one copy, the active copy present in XRQ. And the other observation is that in our case, the higher sucrose concentration, which has the lower osmotic pressure, also corresponds to the higher volume, suggesting an active water transport. So to assess this question, we wonder if the raw sap is involved in nectar elaboration, which is consistent with the presence in the close vicinity of the nectary of xylem vessels, as shown on the fluorescence picture on the left. The blue fluorescence corresponds to xylem vessels. We thus tested the hypothesis of water diffusion from xylem with dyes. Fluorescein is a cationic fluorescent dye that cannot cross plasma membrane. It is distributed throughout the plant by the apoplastic route. When the stem of cut flowers is dipped into 0.1% fluorescein solution, the dye distributes in the stem and seems to accumulate at the very base of the receptacle, as shown on this fluorescence image. Involucral bracts are hardly stained. When facing the head, we cannot see any fluorescein in the ligule or in the petals. We only see the autofluorescence of pollen coats. On sagittal section, we see fluorescein accumulation at the basis of the receptacle and in older ovaries, leading to the conclusion that raw sap is retained in the receptacle until fertilization. We tested 10 genotypes, and we never found any fluorescein in nectar. We did the same experiment with neutral red, which is, as its name states, neutral, and then diffuses freely across membranes and diffuses mainly by the symplastic route, and methylene blue, which diffuses by both symplastic and apoplastic routes. We didn't find any traces of dye in nectar. This confirms that there is no passive diffusion, and that water transport must be active transport, or at least facilitated. But nectar is not only water and sugar. There are many other molecules that we analyzed, but LCMS. So the whole composition of nectar shows interesting variation between genotypes. We found more than 200 species, with a third being unknown. On the right, we can see that each column corresponds to a single molecule that are clustered by class, by molecule family. And on lines corresponds to different samples clustered by genotype. And we can see that genotypes are easily distinguished by the relative amount of different molecules. For example, CI lacks decarboxylic acids, while IR and XRQ have some. The decarboxylic acids, such as suberic and sebacic acid, are molecules involved in plant innate defense by the constitution of cuticular wax. IA also have specifically antimicrobial molecules such as chalcones and benzopyrans. Exarchio have fatty acids and derivatives, but their role in nectar is unknown. The stranger thing is the presence of exotic glucosides in CI nectar, such as 3-ortho-alpha-galactofuranosyl D-mannose, which is produced by Aspergillus. This suggests the presence of fungi in CI nectar. In 2020, Rering et al. Showed that nectar microbiome may affect attractivity. In sunflower, the presence of fungi, the putative presence of fungi in CI nectar and the presence of antimicrobial molecule in IA may affect microbiome, affecting in turn attractivity. So, in conclusion, we saw that corolla shortening is not significant in our 3 lines. Corolla shortening in response to drought stress is not significant in our 3 lines. We saw that pollen quantity is affected by drought stress, but not its quality from a nutritional point of view. And from a genetic point of view, attractivity is correlated to nectar volume, but sucrose might be involved. Sucrose in CI may enhance attractivity. We saw also that there is a probable active water transport for nectar elaboration, and that our 3 lines have different strategies to attract pollinators, which seem related to reproductive strategies. IR produce lots of pollen. Pollen production is weakly reduced by water deprivation, but it produces very few nectar, which production strongly decreases with drought stress. So we can conclude that IR has a male predominant function and maintains it in drought conditions. CI produces very few pollen, and this low production is even reduced under drought, while on the other hand, it produces large quantities of nectar with high sugar concentrations, and it maintains this sugar production high in drought conditions with the presence of sucrose. And this nectar production leads to the conclusion CI has a female predominant function and can maintain it in response to drought stress. Finally, Exarchio produces decent amount of pollen and nectar, but both are strongly reduced in response to drought stress. So we can conclude it has a balance between the 2 functions, but seems unable to maintain them under stress conditions. So the next step of our study will be, of course, to repeat those experiments. And I will also do some RNA sequencing on our 3 lines, which will enable us to identify the nectary identity genes, the secretion genes, and the drought response genes. When these genes are identified, we can do expression polymorphism in the nectary on a hybrid panel. And we will be able also to build gene networks and to establish the correlation between genes, metabolites, and attractivity. And as the question was raised very recently, we also studied the microbiome of the nectar with collaboration through a collaboration with an microbiologist. I would thank the Sunflower Biological Resource Center, especially Marie-Claude Boniface and Camille Tapi, and I would warmly thank the ASTER team and especially Nicolas for his trust on his project, on this project, and Maëlys Bobin, you can see here, who was a bachelor trainee last year. She did a really big load of work, but betrayed us and joined the dark side of the sunflower in Stéphane Mugnoz's team. Thanks for your attention.